Okay, we're going to get started. Welcome to NF2 BioSolutions webinar, Bacteriotherapy for NF2. We are so happy you can join us. I am Helene Bader, a volunteer with NF2 BioSolutions, and I have just a few quick housekeeping notes before we get started. So first, all participants will be muted during the webinar, and you'll be able to ask any questions through the chat function, but we will have the chat box locked until after the presentation, and then we will open it up for questions. If for any reason we are unable to get to your question or we don't have the answer, we will put together an answer document and send that out to all attendees in the next few days. The webinar is being recorded, so that will be available on our website for future viewing. And finally, we would love your input on what webinar topics you would like to see in the future. So please feel free to enter your suggestions in the chat box as well. And now I'd like to introduce our presenters. So Dr. Gary Brenner is the director of the Massachusetts General Hospital Pain Medicine Fellowship since 2002, and he currently sees patients at the MGH Center for Pain Medicine. He is also an associate professor of anesthesia at Harvard Medical School. Dr. Brenner's lab is focused on developing gene and cell-based therapies for Schwannoma and other NF-associated tumors. He has authored more than 50 articles, reviews, chapters, and abstracts on gene therapy strategies for NF tumors, the pathophysiology of pain, stress effects on immune function, and clinical approaches to chronic pain. Dr. Brenner currently holds several national leadership positions related to pain medicine education and training. Dr. John Mekalanos is the Lehman Professor of Microbiology and Molecular Genetics at Harvard Medical School and served as chair of the Department of Microbiology and Immunobiology for 2 decades, starting in 1996. His research spans multiple facets of bacterial pathogenesis with an emphasis on using genetic and functional genomic approaches to explore virulence gene regulation and host pathogen interactions. His laboratory has provided many key genetic tools that have been successfully used in the field for decades and have defined the mechanisms that have led to bacterial virulence. Dr. Mekalanos has received many honors, including election to the National Academy of Sciences and American Academy of Microbiology, the Eli Lilly Award, City of Medicine Award, the Sanofi Institute Pasteur Award, and the Drexel Prize. He has been a member of the FDA Advisory Committee on Vaccines and Related Biologics and has consulted for numerous governmental and private agencies. We are thrilled to have both Dr. Brenner and Dr. Mekalanos with us today. Dr. Brenner?
Thank you for the kind introduction. On behalf of John and myself, I want to thank you all, NF2 BioSolutions, for the opportunity to present what I really genuinely feel is possibly the most exciting project I've ever worked on in my laboratory career. I'm going to try to keep the talk quite conceptual instead of digging too deeply into the data. I will mention this is the first time I'm publicly presenting these data from our lab and the collaboration with Dr. Mekalanos. So I always like to give a little bit of history to provide an underpinning for why we're doing what we're doing. Almost nothing that we do in science is something that isn't in part based on the observations and work of people who came before us. So there actually is a fair history of what's come to be known as bacterial cancer therapy. In 1725, Antoine Didier, a French physician, observed that tumors in patients with syphilis seemed to be cured more frequently, and syphilitic prostitutes appeared to have lower levels of cancer than the general population. About 100 years later, an English physician, James Paget, suggested that an infection in one of his patients may have caused tumor regression. And then about the same time, a German physician, Wilhelm Busch, found evidence of cancer regression following a post-operative infection. Now, these reports weren't widely disseminated, and the watershed moment was really occurred with an American physician, William Coley. And he was an oncolytic surgeon. And in 1890, he treated a 17-year-old Elizabeth Daschle for sarcoma of the limb. The standard of care at the time was amputation of the affected limb. Unfortunately, Ms. Daschle died 10 months later from the same cancer, which had metastasized. And as with many treatments today, which involve resection, unfortunately, the results are not what we would hope for our patients. After this experience, he reviewed New York hospital records and found a case of one patient, a Fred Stein, who had inoperable neck sarcoma that absolutely vanished following a bacterial skin infection with the gram-positive streptococcus. He was so interested in this that he dug deeply into the literature, which wasn't easy at the time because there were no computers. Reminds me of my PhD training. Extensive research into the medical literature. And this is why I mentioned those other physicians, Didier, Paget, and Bush. And he saw their reports. And that, in conjunction with Fred Stein and 47 other cases of infection associated with cancer regression, really got his interest. And he made this his life work. He proposed, and really the first person to widely disseminate this concept, that bacterial-based cancer regression was, in fact, something that occurred not infrequently and the result of actually anti-tumor immune responses. Imagine that. Prior to knowledge of the cells of the immune system, he made this amazing hypothesis. And in 1891, he did something which naturally could not be done today. He injected an inoperable throat tumor with streptococcus, and the tumor resolved after multiple such injections. Back in those days, people often experimented on themselves, in fact. And in 1993, he did what was critical. He had an observation, he did some research, he proposed a hypothesis, he tested the hypothesis, and then he published. So he really did what any good scientist should be doing today as well. And he published treatment of malignant tumors by repeated inoculations of erysipelas, which is a skin infection, with a report of 10 original cases. And he devoted his life to developing a bacterial cancer therapy. Unfortunately, our understanding of bacterial biology, of the immune system, of human genetics, obviously was in its infancy, and he really never got to a truly effective treatment. But much more recently, all of those factors have changed. In 1990, the FDA actually approved use of live bacteria, BCG, for non-invasive bladder cancer. And that treatment remains the standard of care today with a reported 50% cure rate. It's quite amazing. And the treatment is thought to represent an immunotherapy, as these bacteria are highly immunostimulatory. The product, unfortunately, is produced only by Merck, and there's currently a worldwide shortage. And it should be used with 6 applications, but frequently now it's used in 3 applications because of that shortage. Since approval of this bacterial product, BCG, for bladder cancer, there's been increasing interest generally in bacterial cancer therapy. This has been occurred because of development of genetically modified bacteria that can improve both safety and efficacy of the treatment. There's a large number of animal studies that have used many different strains of bacteria and have shown value in treating multiple different cancers. Of course, these are animal models of cancers. There are, I'm aware of at least a few biotech startup companies that currently exist and are devoted to developing bacterial cancer therapy. And there are actually multiple clinical trials currently underway, though unfortunately, none have yet led to another FDA-approved bacterial treatment for cancer. But I do think that's on the horizon. Now, this is the key point and one of the insights that we're bringing to the table. Bacterial therapy, as far as I can tell, and I've looked deeply, has never been proposed and obviously never tested for the treatment of any benign or non-cancerous tumor. Obviously, Schwannoma is a benign neoplasm in virtually all cases. So that's the history. That's the current update. Let's talk a little bit about why we think bacterial therapy for Schwannoma and related NF tumors makes sense. So how does bacterial cancer therapy work? There's direct killing of tumor cells. These bacteria can actually get into the tumor cells, and they have innate mechanisms that allow them to kill those tumor cells. They also inhibit angiogenesis, which is they deprive tumors of their vascular blood supply. And without the blood supply, these tumors don't grow. As you know, the only drug that's really routinely used for Schwannoma now is bevacizumab or Avastin, which inhibits angiogenesis. And finally and amazingly, these bacteria have been shown to initiate anti-tumor immune responses. So they turn the host's immune system on and allows it to attack the tumor cells. So specifically for Schwannoma, why do we believe that it makes even more sense, actually, than cancer therapy? Well, Schwannomas are solid tumors with a hypoxic core, and these bacteria, the ones we're using, thrive in a low oxygen or hypoxic environment. The tumors are slow growing. So unlike cancers, these bacteria will have more time to replicate within the tumor and attack those tumor cells, and more time to develop an immune response to attack the tumor cells before they can change. And they're highly vascularized. So for many reasons, we think this makes sense. And one key point is that Schwannomas are known to contain the immune system cells that are necessary for the bacteria to induce anti-tumor immunity. And I'll just tell you one other piece, which is we think that Schwannomas are what we call immunologically cold. There's a lot of suppression of immune responses, unlike cancers, which are hot, immunologically hot, a lot going on immunologically. And we believe, and we have data to support this, that bacteria have the capacity to change the Schwannoma from immunologically cold to immunologically hot and cause almost a vaccination-like effect, allowing the host to start killing the tumor cells with immune system mechanisms. So just to recap, bacterial tumor therapy, it's rational for Schwannoma treatment. We plan to deliver via direct tumor injection. That's a minimally invasive procedure. These tumors can be readily localized via MRI or ultrasound. And if I didn't make this clear, these bacterial products have been used in humans, they've been used safely in humans, and they've even been used in humans via direct tumor injection. As I mentioned, Schwannomas are slowly replicating and genetically stable, and we believe this will enhance this therapeutic strategy. And as I mentioned, they're hypoxic, which means a low oxygen environment in which the bacteria can thrive, and they're very vascular, which is another feature of the tumor that the bacteria can attack. Our bacteria, again, has proven safe in multiple clinical trials. This is critical. So we only minimally need to worry about whether we're going to harm patients with the therapy. I think that's very, very unlikely given the clinical experience. And tumor killing occurs via multiple pathways, and importantly, including induction of anti-tumor adaptive immune responses, so specific immune responses. And I'll give you a little bit of a punchline because I'm not going to show you these data. I'm going to try not to make you punch dumb with data today. As I said, make it more conceptual. But we've shown in the laboratory that treatment of a single tumor with direct injection of the bacteria controls growth of a tumor in another part of the animal. Secondarily, we have shown that treatment of a single tumor allows later on control of a developing tumor. So if we treat one tumor, wait, and then implant another schwannoma, the second schwannoma, which hasn't been directly treated, is actually growth controlled better than the original tumor. It's quite amazing. So I know many of you know these materials I'm about to present in this slide back and forth. I don't mean to speak under anyone. I just want to make sure everyone on this call has the necessary background related to schwannoma. Schwannomas, as I said, are non-malignant tumors that grow in peripheral nerves. But benign, I think, is a really bad word. I like non-malignant because they're anything but benign, particularly because they grow within the cranium, within the skull, and in the spine, and do horrible damage to patients. They appear largely first in children and young adults, with new lesions occurring throughout life, further increasing the burden of disease. Multiple lesions are generally or essentially always present unless it's sporadic schwannoma. And as I've alluded to, the tumors cause debilitating neurologic disorders, including severe persistent pain. I became interested in this field because of my treatment of patients with pain. I work solely in a pain clinic, clinically. And there's substantial psychological comorbidity, as you know, not just for patients, but for their family members. And the current standard of care, resection, usually operative and to a lesser extent with radiation or radiotherapy, is itself associated with significant damage to the nervous system. And some of these tumors, because of their location, can't be removed surgically because it's just too dangerous. So, in order to test our hypothesis or our idea that bacterial therapy for schwannoma makes sense, we needed animal models. And unfortunately, there aren't a lot of animal models for schwannoma. We actually developed two in order to test the therapies, the gene therapies and this bacterial therapy we're developing. And what we do is we take human NF2 schwannoma cells and directly implant them into a peripheral nerve of a mouse. And you can see that here. And this is just a glass catheter we use to inject the cells. And the cells grow just fine on their own. We've also genetically modified the tumor cells so that when we add luciferase, the cells light up, and with a very sensitive camera, we can actually see basically how many cells there are. And here are two animals with very few number of tumors, and here are two animals with a large tumor burden. So we can follow animals across time while their tumor is developing and do a treatment, in this case, inject bacteria directly into the tumor and see what the effects are. So these were the first data we obtained, and it was kind of one of these, oh my gosh moments, like, is this real? The red line is the control injected tumors. These are 8 animals in each group. And the green line is the bacterial injected tumors. And what you can see is there's an unequivocal effect that's lasting as well. We are developing a gene therapy, which I'm already very much excited about, and we're trying to get it to clinical trials. I'll tell you, we never saw anything of this magnitude. And you always have to repeat your findings. And this was our second experiment, and we exactly repeated the findings of the first experiment. We do these experiments in a way that's called blinded, so we don't know which animals are which. That's to remove any bias. In 3 independent experiments, we found that 75% of the bacterial injected tumors fully resolve. They just melt away. This is a very unusual finding in the bacterial tumor therapy literature. We wanted to look at a second model in the cancer therapeutics field. One model is always considered inadequate, and I agree with that. So this is a completely different model. It's a different mouse, and in this case, we implant a mouse, not a human, NF2 tumor line. And we find that these cells grow a lot faster. You may not have noticed that before we were looking at weeks on this axis. Here are days, but we see essentially the same effect. The control animals, their tumors grow and grow and grow. In fact, we have to sacrifice them because the tumors get so big and it affects their nerve function. But the bacterial treated animals, the tumor burden is kept much smaller. Very exciting data to us. But we need to know how it works. We need to have mechanism. I'm going to run through these data fairly quickly. If someone had real interest in seeing the data in depth, I'm happy to share it. We are about to submit our first paper for publication. First, we found that bacterial injected tumors undergo what we call apoptosis or programmed cell death. They actually undergo several types of cell death, but certainly apoptosis. And here you see bacterial and control treated tumors. These are slides showing this, and a stain for apoptosis. And you see a lot in the bacterial treated tumors, really a tremendous amount, and very, very little in the control samples in both the models. We also find in the bacterial treated animals, there's inhibition of blood vessels. And here are the control treated tumors. And trust me, when you look at them under the microscope, they're big, red, and full of vessels. And we see a lot of vessels with staining. And here are the bacterial treated tumors, shrunken, white, and very little evidence of vessels. It's really exciting. And it's what, honestly, it's what we would have expected from the bacterial cancer literature. There's also infiltration into the tumors of immune system cells, both lymphocytes and macrophages. I won't belabor the staining, but there's a great increase, I'll go into that in a minute, in the resident immune cells. And we think that's very important for generating the immune response. Perhaps even more interesting than the increased number of immune cells is what I'm about to show you. Macrophages, which are key cells in generating immune responses, come in two flavors inside of tumors. One is tumor promoting, they're called M2, and the other are called M1 or tumoricidal. The names are obvious. Resting schwannomas, that's untreated schwannomas, seem to have a predominance of M2 type macrophages. After bacterial injection, the green bars, here's day 3 and day 7, what you see is a shift in the control, red, from M2 to M1 tumor-killing macrophages. This is a very exciting finding because these are key cell types. Some of you, I'm sure, are familiar with different kinds of lymphocytes, which are what we call the effector cells in these processes. And as I mentioned, schwannomas have lymphocytes. Here's the control, red bars. Unfortunately, in the untreated tumors, as in humans, most of the lymphocytes, there are not many killer lymphocytes that would kill tumor cells, but there are a lot of what are called Tregs or regulatory T cells that actually stop the body from being able to generate anti-tumor responses. Now, after bacterial injection, green bars, an amazing thing happens. You vastly increase the number of tumor-killing T cells, and you decrease the regulatory T cells. So exactly what you'd want to see if you were designing a therapy. And I'll tell you, we also see increase in something called helper T cells. And they're another lymphocyte type that can facilitate immune responses. It's always important to look for toxicology or damage from any treatment. And I'll just tell you that with very sensitive sensory testing in these animals, we see no effect of bacteria on the animal's behavior. And we also see no effect on motor function. So these data in animals mimic what we expect to see from human data, which is these bacteria are safe. They don't cause problems on their own. Naturally, we have to think a lot about the tumors in the spinal cord or in the cranium, in the skull, but we do think there's ways we can design these bacteria to be so safe that it will even make sense to inject them directly into tumors in the cranial vault. So, a summary: bacterial tumor injection of schwannomas, a novel schwannoma immunotherapy. Safety's been established in early phase clinical trials, 2 intravenous, 1 direct injection, and actually 1 oral, with exactly the product we're using in our animal studies. We've shown efficacy, i.e. it works in 2 completely different schwannoma models and replicated the findings. We've shown that there's tumor killing via apoptosis, programmed cell death, vessel growth control, and most important, what we call immunogenic cell death, which is a form of tumor cell death that causes anti-tumor immune responses. And these are the shift through tumoricidal macrophages from tumorigenic macrophages, increased cytotoxic and decreased regulatory T cells. And these, we believe, are what are leading to induction of host anti-tumor adaptive immune responses, what we would generally call a vaccination effect. And this has the potential to control growth of multiple tumors following bacterial treatment of a single schwannoma, and the potential, we hope, for lasting anti-tumor immunity following treatment that could control the growth of subsequently arising tumors. I think now you know why we're so excited about this. And I haven't shown you this today, but we've enhanced the effect of the bacteria on anti-tumor immunity with use of immune checkpoint inhibitors. So I don't want you to think this would only work for schwannomas, because NF2 patients have other types of tumors, and we also care about other benign neoplasms, obviously. And I will just briefly show you that we have efficacy data with the same product in benign meningiomas, and the effect is really quite obvious and lasting. This is 41 days, which is quite a long time in animal studies. And we even looked at a NF1 associated tumor. This happens to be a malignancy, a peripheral nerve sheath tumor. But again, we see a quite clear effect on tumor growth. So this adds to our excitement in the project. And while we're using an existing bacterial product that may in fact work very well in humans, we at the same time are working on ways to enhance bacterial-induced schwannoma and other tumor cell killing, because we know these products haven't worked in human beings for cancers. Again, schwannoma's fundamentally different as in meningioma, but we want to more or less hedge our bets to get to a real treatment. So what are we doing? We're re-engineering these bacteria to enhance anti-tumor immune responses. So we can engineer superadjuvants into bacterial cells. There are a variety of types. And what I'm showing you here is something that's relatively recent out of the lab, where the bright white dots are actually our bacteria that we've re-engineered to express a protein called GFP that lights up. And what's really amazing is we kill all the bacteria that are outside of the tumor cell. So everything you see are bacteria inside tumor cells. And maybe you can kind of see this is a tumor cell and it's chock full, 4 bacteria in there. Here's another tumor cell with what looks like 4 or 5 bacteria inside. And I'll also say we've engineered this protein, GFP, to be secreted by the bacteria. It's very exciting. So we're at the same time, actually right now, waiting to hear from someone. We're working to engineer the bacteria to express molecules that would specifically enhance anti-tumor immunity. It's all very exciting. And working with John's lab, we want to eliminate bacterial properties that might inhibit development of anti-tumor immunity. At this point, I want to turn the talk over to John. But before I do that, I just want to express to the audience how excited we are to have John as part of our collaboration. You heard the intro, but I don't think it really gives you the true flavor of the fact that John is a giant in the field of bacteriology, and I couldn't imagine a better person to work with to help us re-engineer these bacteria. With that, I'll turn things over to you, John.
Okay, thank you very much, Gary. I really appreciate it. And I want to emphasize again, despite the accolades and compliments, I've never worked on anti-tumor bacteria, if you will. This is a new area for me. I feel like my whole life, 40 years plus of research, has been devoted to understanding how bacteria interact with cells in the immune system, and we've made some pretty important contributions along the way. But when I was approached by Gary and his colleagues to consider getting involved in this collaboration, I was seriously very excited to see these data and realized, wow, this is just perfect timing because we have a lot of new technologies that could potentially improve the potency and the efficacy of this bacterial-based therapy for Schwannoma. And Schwannoma was, as I learned, was such an unmet medical need. The field largely dominated by all kinds of other inoperable neoplasma of so many varieties that aren't responding to checkpoint blockade or aren't responding to other therapies. So I think, you know, having a chance to sort of hone in on this problem with Gary's guidance and fantastic animal models and fantastic preliminary results, it was just an opportunity I couldn't pass up. But as I said, I don't work on the tumor models in general in my laboratory. So my focus of making a few comments is just going to be to try to give you some excitement about where we are in the bacterial pathogenesis field, because we feel we're learning so much about how bacteria interact with cells in both positive and negative ways, if you will. Positive being kill a cell, but you know, negative can be anything from causing host damage to suppressing an immune response. So all these things are important when you think about a bacterial-based therapy. You don't want the bacterium to interfere with the immune response, but you may want the bacterium to be more potent at killing the tumor cell. And if I don't remember to say it, I'll say it now once, so I've said it at least once. There is an emerging strong literature that says that when bacteria kill a tumor cell or a mammalian cell, that alone can be very immunostimulatory. As Gary pointed out, the Schwannoma cells can undergo apoptosis. There are other types of cell death, myriad names, necrotic cell death, pyrotosis, etc. And we're starting to understand the molecules that drive cell death in different directions. And those cell death pathways actually have tremendous consequences for the immune response of the antigens that are released, which would include tumor-specific antigens. So with that said, now I feel better. I won't forget to say it at the right time. So Gary, if you could change the slide. So where we're going to focus is we're going to focus on enhancing direct tumor killing by bacterial cells. And we have a variety of different ways we can go about doing this. One of the primary ways that we're very interested in doing is we've devised methods in the lab to allow bacteria to target specific cells. These are genetic methods where we can find the needle in the haystack, and we're really excited to apply this methodology to bacteria. First, the bacterium the strain that Gary has the most experience with, but eventually a completely re-engineered strain, because I think there are advantages in that as well, and target those bacteria to Schwannoma cells with very specific genetic methods. Our prediction is that that's going to improve the specificity, perhaps even the potency of the bacteria in the therapeutic models that Gary's devised. We'd like to improve invasion of tumor cells. Certainly these bacteria invade the tumor cells, as Gary pointed out with the GFP image you see up at the upper right-hand corner. But it is possible that we might be able to get quantitatively more invasion by decorating our bacterial cells with molecules that specifically bind Schwannoma cells. And we also feel that invasion is an interesting property. Bacteria have multiple different ways they can enter inside a cell, and some of those pathways are immune suppressive, and some of those pathways are immune stimulatory. So by engineering exactly the right invasion pathway, you may also, again, greatly stimulate the potency of the bacterium for both killing the tumor cell, but also activating the immune system. And we also are very interested in all kinds of virulence factors, but I use just this one because you'll see some beautiful videos in a second. Bacteria produce very powerful cell-killing machines, and we call these secretion systems because they literally take proteins and inject them into a target cell, in this case a tumor cell, using a nano machine. Think of it as a very small syringe, just nanometers in size, really, 1/10 or 1/100th the size of a bacterial cell. So with these very powerful little syringes, if you will, bacteria can inject very potent killing toxins, if you will, molecules that have been designed or otherwise are known to kill cells in a very specific fashion. And we know a lot about how to do that and have designed these systems from the ground up. So on the next couple of slides, I'm going to show you examples of how phenomenal these killing machines are. So what you're looking at right now is the red cells. Just think of them as good cells. The red flashes you see are one of these nano machines, these secretion machines called type 6. The dark cells, if you will, are bad cells. They happen to be E. coli, but in this model, you can see the red cells are firing away their little machines, and watch what happens to the E. coli's. Goodbye. So bacteria can use these machines to kill other bacteria that they're competing with, and that's interesting ecologically. But it has applications, and we've already developed one application where we're using a good bacterium to protect animals that are commercially important in the food industry from bacteria that are bad, using these kinds of killing machines. So this idea has been kicking around, but now we're really starting to use it for applications. We'd love to be able to do this specifically with tumor cells, specifically Schwannoma. So next slide, please, Gary. So the next video you're going to see gets a little busy after a while because the bacteria are growing while everything's going on. So I just want you to briefly focus in on a couple of things. First of all, the big cells you see on the field, painted or arrowed out in red. Yes, exactly, Gary. Those are tumor cells. And the tiny cells you see with the white arrow up at the top, those are bacterial cells. And you can see tumor cells are literally 1,000 times the volume of a bacterial cell. But we've been able to show that even a single bacterial cell can kill that tumor cell using these kinds of machines. What you'll see is also a phenomenal thing that goes on now is that the bacteria that we're going to use to attack these cells have the machine marked with a protein called green fluorescent protein. So the bacteria will turn green under a fluorescent microscope when they turn on the killing machine. And what is really amazing about this is the bacteria are so smart already that they don't turn the machine on right away. They have to bind to the target cell. Binding is enough to activate a pathway that the bacterium senses and says, now it's time to turn on my killing machine. And that's going to be indicated by green cells. And you'll see that the green cells that are very close, if not stuck to the tumor cells, are the ones that do the job. So if you'll run the video right now, you'll see it in action. This is time lapse, so a picture's taken a little herky-jerky every 10, 15 minutes, as I recall. A snapshot is made, but it's turned into a video. So you saw where the bacteria were. They're merely growing and binding to cells. They just keep on going. And then all of a sudden, some of the ones that have bound start turning green. And you can see if you look across the whole field, there's lots of bacteria, but where are the green ones? The green ones are right next to the tumor cells that are no longer tumor cells, okay? So that's the amazing thing, is that what's left here are just bacteria stuck to what are called cytoskeletal components. These are the leftover dregs of what is more or less a vaporized tumor cell. We can imagine that when that tumor cell lysed, all kinds of immunostimulatory molecules were released from the target cell. But very importantly, this demonstrates that it may even be possible to very specifically kill tumors without the bacteria ever even entering the tumor cell. And that might have consequences for the type of immune response you get. So both invasion, being able to make the bacteria enter tumor cells more efficiently, as well as preventing invasion but still killing tumor cells, it needs to be investigated. And I think we have the preclinical models through Gary's collaboration to really tease out, at least in the mouse model, which of these properties correlate best with immune responses. And if that adds to our information on how to devise a better human therapeutic agent, we will be delighted, because this is, we seldom have a chance in our basic research to have such a great shot on goal here for applied science. And I think we can get this into the clinic pretty quickly using the models that Gary has described. So I'll stop there and pass the ball back to Gary to finish up.
Thanks, John. It's a pleasure to work with someone as humble as you. What you failed to mention was you discovered one of these key bacterial mammalian cell killing machines, and it's really an honor to work with you. I mean that.
Well, thanks.
So I want to give some credit to Sharif Ahmed, who's the key scientist in my laboratory, and it was really Sharif and myself who came upon this concept of bacterial therapy for Schwannoma and other benign neoplasms. And I'll just mention that it was a direct offshoot of the biology of the gene therapy we're doing. So they are related. But I don't believe any great discoveries or work occurs without collaboration. I welcome collaboration. Personally looking for what John alluded to, which is to translate work from the laboratory to a treatment which helps people. My life goal, and I'm sure John would say the same, isn't to better treat disease in mice and rats. It's to find something that helps patients. It's a great need. I will also mention that we have other members of our collaborative work. I work with a tumor immunobiologist. She's wonderful. Her name's Xinhui Wang. And I work with a number of surgeons, because as you'll hear in a moment, we want to look at human tissue as soon as possible to help us better design the next therapeutic, the improved bacteria that John discussed so eloquently. So what are the next steps? As you heard, this is really currently getting started, but it'll be the major push in the near future, which is genetic engineering and animal testing to develop more effective, and I would add safer, bacteria with fail-safe mechanisms, but better strains at the end of the day. We want to investigate the biology of these improved bacterial products in fresh resected human schwannomas. The fact is there's only so much you can learn by looking at these animal tissues. There are fundamental differences both between the tumors and the host immune responses in animals and man. And in order to be smart and make the right product that has the highest chance of being effective and the highest chance of being safe, we need to look at human tissue as soon as possible. And we hope to use those two, the information from those two above, to obtain the large amounts of money through federal funding to move to early phase clinical testing with the current product, use the information. I mean, if it's a home run and it happens to work and it's safe, no one would be happier than I. But I think we have to go into this with the understanding there's a good chance the current product might not be optimal, but we can use that information from those studies to help us design the product that will be the therapeutic, we hope. I think that's really all I have to say. John, do you have any summary comments?
No, I think you echoed my feelings exactly. I think we all know phase 1 human testing, development of a product for real, to have it on the shelf and available for treatment. It's going to take more data and more funding, but we are well on the way, I think, in this collaboration to being able to generate both of those with a little bit of luck.
Great. And at this point, I'll turn things back to Helene, I believe.
Yeah, so thank you both for such an amazing presentation. That was wonderful, and this is really exciting research. We do have the chat box open for questions, so please feel free to enter your questions there, but we do already have a few questions for you. So we'll just get started and dive right in. The first question is, you mentioned 75% of bacterial injected tumors fully resolved. Did the treatment shrink existing tumors or stop and slow the growth of new tumors after inoculation?
That's a great question. So in the human tumor model, they actually shrunk and made the tumors disappear. There was no evidence of tumors in 75% of the animals. There was no signal. And when we look at the nerves, they look quite normal. In the mouse Schwannoma model, the tumors, some shrunk and some slowed. The overall effect, when you look at averages, was decreased tumor size. But we didn't see those tumors completely go away. Interestingly, in what we call, in that same model, the re-challenged tumors. So that's where we have one tumor growing, we treat it, we wait, in this case, about 2 weeks and implant more tumor cells. Half of those tumors completely resolved. So that's a great question.
Great. We also got a comment. This has been a fascinating talk. Thank you. And a question, can you please discuss more the situation involving the stop break on the immune response? If the immune response is not stopped, then autoimmune and inflammation results. Where is the science at as far as this is concerned, and how difficult will it be to put this sort of treatment into someone without the proof that the response can be shut down?
Sure. So the fact of the matter is, we don't know at this time which antigen or antigens, tumor antigens, in our animal models are the basis for the anti-tumor immune response. Having said that, we don't see any evidence of either motor dysfunction or sensory dysfunction. And with the sensory tests we are using, we can see what we call gain of function and loss of function effects. So pain, for sensation, we could see pain, we could see decreased sensation. So at least in the animal models, we don't see the development of autoimmunity. Having said that, it is an important consideration. In the bacterial cancer therapy trials that have occurred thus far, I'm not aware of any cases of autoimmunity occurring. It is possible, and what we would hope is that the immunity is based on a tumor-specific antigen. In this case, it would be unlikely to develop autoimmunity. But again, I want to, say to the person who brought this up, it is a very important consideration. And in developing clinical products and testing them, one of the most important consideration, if not the most, is not making the life of the person you're testing something on any worse than it already is.
I think Gary did a great job of answering that question from the point of view of autoimmunity, perhaps driven by now a new immune response against the Schwannoma antigen. Obviously, we wouldn't want B and T cells running rampant and attacking healthy Schwann cells throughout the body. But the other part of the question I think that I also picked up on is really the immune checkpoint effects. And Gary has very convincing data that one type of immune checkpoint blocker, if you will, an antibody that blocks a very critical molecule in the checkpoint response. I think it's PDL or PDL1. But anyway, it's being used in many cancer treatments successfully. It seems to enhance the anti-tumor response. So I would say from that point of view, it sounds to me like it's classic checkpoint blockade controlling a very, potent immune response against the Schwannoma. But even with that, Gary, those experiments showed no toxicity, no autoimmune, no systemic autoimmunity, as best you could tell. So I think that for transient treatment to block the checkpoint is one example of sort of an adjuvant therapy of trying to enhance the immune responses. Most of the molecules we envision are not going to be chronically on board. They're going to be transiently used in the context of the bacterial therapy, and eventually they will go away. And we certainly don't expect any worse toxicity than folks are seeing with these molecules in standard cancer. Not that that is, some of them have toxicity problems, like CTLA4 you can look up, has a lot more toxicity associated with it, but does a lot of the same stuff. So I think we need to investigate that when the time comes. But Gary, perhaps you can go one step further and say, I mean, that would be kind of the next phase. We have a successful therapy working pretty well, in phase 1 or phase 1A, B, extended studies. Yeah, you can take those checkpoint molecules right off the shelf and combine them. So I think the follow-up clinical trial could happen very, very quickly.
Yeah, thanks, John. And that's certainly part of our plan. If it's necessary to use checkpoint inhibition, we would roll that into a clinical trial. The human schwannomas do have PD1, PD-L1, and CTLA4 based signaling. That's what the evidence would show. We have some data to show that, which is why we modeled that in the mice. Fortunately, the mouse model also has the PD1 pathway. And one advantage there is if the checkpoint inhibition is necessary for efficacy, if there were autoimmunity, it is possible that removing those molecules from the system would deal with the autoimmunity as well. So we are definitely thinking, or doing our best to think a few steps ahead. And as John alluded to, combination of cancer therapeutics with a standard agent and a checkpoint inhibition is really very common. And I suspect that might be the direction we eventually go.
Great. We have a bunch of questions, so we'll try to get to as many as possible. We have a comment, really cool project, and then this is a multi-part question. So how many times were the mice treated, and were the bacteria directly injected into the tumor? Did you also try systemic treatment, and did you look at the lifespan of mice with the mouse tumors that were treated with the bacteria?
All good questions. So the tumors are injected once directly, and we actually have two different tumor models, even with the mouse model. One is the cells are in the nerve, and I didn't mention this, but the other is a subcutaneous tumor to allow the animals to survive longer. We haven't conducted a survival study yet. It's something that we may do at some point. I don't know if it's truly valuable in the context of the model systems we're using. The effect size in terms of tumor regression or tumor control is so great that I'm just not sure what value in animal models would be derived from survival studies, though, as the questioner rightly points out, that is a very standard question that's asked in preclinical cancer models.
And have you ever contemplated using another animal model that may be closer to a human, like a swine?
Yeah, so the swine, of course, yes. The swine, NF2 swine, as far as I am aware, are not yet fully characterized and not yet available for preclinical testing. I would love to try it in one of these animals when they become available. I will mention the pigs cost about $10,000 a piece. There's an NF1 pig, as you probably are aware, and then animal housing becomes more expensive. I do think it has value, but on the other hand, we have a bacterial product that's been repeatedly shown safe in human trials. I really think it probably makes more sense to skip large animal models and go right to humans if that's possible.
Okay.
I think there was one other component of the previous question that I missed. Is that correct?
The lifespan of mice with the mouse tumors.
Was there something before that?
And did you try systemic treatment?
Yes, that's right. So we haven't utilized systemic injection. That has been used in humans and mice, as we've alluded to. There's a reason for that. It may be that part of the reason that these treatments failed in human trials is that the tumors weren't colonized with the bacteria. Human beings seem to be quite a bit better at clearing these bacteria, the ones we're using. Obviously, I'm being a little discreet about that. That won't last long. Because the human immune system clears them better. So my thinking was it would make more sense to use a direct tumor injection that allows a higher inoculum or higher number of bacteria to be injected and thus increase the likelihood of colonization. And there are some published human data that would very clearly indicate that that's the case. I will depend on John to think about and help us engineer bacteria that are more capable of colonizing tumors without increasing the likelihood of causing systemic infection and significant toxicity to the patient.
Mm-hmm. Can you share what the bacteria used in this research was and why you chose it?
I can share why I chose it, which is a large preclinical body of data that indicate these bacteria can kill by all the mechanisms we discussed. A variety of forms of cell death, including immunogenic cell death that leads to anti-tumor host immune responses, control of the blood supply, and changes in the innate immune cells in the tumors. The reason I'm not sharing the exact product is because we do have a patent submitted. It should be published within a couple months. Once that occurs, I'll be free to openly share the exact product. As I mentioned, we're very close to submitting the first publication on this, which would obviously include the product. The other reason I chose to use this bacterium, which I think we've discussed several times, is it's been what we call de-risked in clinical trials. That is both by intravenous injection and direct tumoral injection, it can be used safely.
Great. So in the interest of time, we do have a number of questions that will go unanswered right now, but we will definitely reach out and be able to answer these later. But one question that keeps coming up, and I'm sure you know what it's going to be, but it's basically the question in different ways of asking, what will happen next? Are there clinical trials happening? What's the biggest roadblock to getting it to clinic? How long will this take? What do we need to do to get this moving along? Everybody's very excited for this. So I wanted you to be able to address that, and then we can have our next presenter come on to talk a little bit more about what's next.
So the fundamental roadblock to clinical trials is funding. NF2 BioSolutions, Nicole, and several of her colleagues, some of whom are on the webinar today, have done an amazing job of hooking us, the scientific researchers, into a variety of resources. Surgeons across the country, FDA experts, and other important resources that are helping us on this journey to develop a better product and get to clinical trials. I think we have all the pieces we need right now. And, you know, I'm sorry to say this, but the piece that's lacking is the funding.
So with that, we are going to actually turn it over to Mike Halloran. Mike is a volunteer for NF2 BioSolutions and the father of 15-year-old twin daughters with NF2. And he has been in the professional development field for 30 years as an executive in the for-profit and nonprofit space. So we'd love to hear from you, Mike.
Great, thank you so much. And I'll just take a minute because I know we're right up against the 1 hour time frame, but I don't know how about all you feel, but is that exciting or what? I mean, just as a parent with daughters with NF2, to hear the results from Dr. Mekalanos and Dr. Brenner are really exciting. Seeing those green bacterial eating the bad ones, I told Dr. Mekalanos that's like a mic drop. I mean, the possibilities from this bacterial research is just tremendous. So my wife and I are delighted to be volunteers to help out Nicole in the NF2 BioSolutions just as volunteers. And the question the next steps are to fund this next trial. And it's $100,000 to take this to the next trial and be able to fund the next trials that will then allow them to take it so the preclinicals allow it to take it to the final clinical and then the human trials and hopefully leading here. So we've set $100,000 to raise through the course of the summer. We're calling it the Axe Out NF2 campaign. Raised will go direct to this bacterial tumor therapy research to take it to the next phase. And we're really excited about it. And we think we can do it. We need support and help, advocacy. So really asking all of you, I know there's 85 on this webinar, to do three things really. One, to be an ambassador and advocate for this research for NF2 BioSolutions, what we're doing, because what we're doing for NF2 tumors, as you heard from our two physicians, can really have application to other rare diseases. And I'm learning all of this and it's really exciting. Second, is to consider a financial investment in this Axe Out NF2 campaign. And we want to raise right off the bat $50,000 and use that as a challenge match to then go to a broader public and do a dollar for dollar match here in July and August. So really focused on first reaching that first $50,000. And then the piece is to, as an ambassador, but refer us, let us know if there's others that should hear this story and might be interested in supporting this campaign and this research. On your screen there, you see our little visual for our campaign, as well as the NF2 BioSolutions. There's a lot more information on all at NF2 BioSolutions, and Dr. Nicole Harwood, who founded it, she's a physician, she's amazing, and her board, the work that's being done there. And then our campaign for Axe Out NF2, there's a web link there. Also, I'll just give my email, and maybe this could be typed in and sent out as a recording, but it's michael@nf2biosolutions.org. So if you've got a direct question that you want to ask about your support or advocacy, you can email me directly. So thanks for letting me take a little bit of time at the end here.
Well, thank you. Thanks so much to all of our presenters and for everyone attending. And we will send the recording out within the week. We'll also send a transcription out. We are sorry we couldn't get to everybody's questions, but we wanted to respect everybody's time. So we will go through and make sure all those questions are answered, and we will send out a document so that no question will go unanswered. Please remember to visit www.nf2biosolutions.org. Email us if you have any questions, and please donate to this incredible research if you can. We thank you for your interest and your contributions, and have a wonderful evening.
Thank you.
Very much. Thank you.
Thank you. Good night.
Good night.
Good night.